questions on the ATP-PC system before we look at the adaptations to training? Adaptations, and then we'll look at glycolysis, which is the second anaerobic system. Okay, so adaptations to training are typically positive, right? It's the body's response to stressing it, and that is usually going to make the individual better at the thing they're stressing the systems with. So you have to make sure that you pick the appropriate stressor for the adaptation that you're looking for. But here in the ATP-PC system, it's relatively simple. So we would maybe expect that changes in ATPase or creatine kinase might produce more ATP, or that the amount of ATP or phosphocreatine might result in more ATP production. All right, so for the ATP-PC system, it's looking for how do I benefit short-term, high-intensity, very short duration type events, right? Can I produce more power, for example? Well, power would involve strength and speed, and those would both involve ATP, okay? So when we look at the research, we have some differences of opinion in the research. It's quite equivocal. So we do see some studies indicate that there's a change in the activity level of the major enzymes, particularly creatine kinase, which is facilitating the breakdown of phosphocreatine, remember? And there have been some studies in resistance type training and also sprint cycling training, and those studies have indicated this increased activity for creatine kinase. So then that ought to lead to faster regeneration of ATP. Okay? So we see that in training, there's some disagreement over whether or not that translates into actual increased performance in the competitive event. All right? If we look at levels of ATP and phosphocreatine in the cell, then again, there's some studies that suggest that ATP and PC concentrations can increase. So when the athlete is at rest, they have higher levels of ATP and phosphocreatine floating around in the sarcolemma, right? Sarcoplasma rather. And that would occur in response, that would occur in response to sprint throwing, cycling, these anaerobic high-intensity events. So when they look at endurance training, they don't see any changes in ATP and phosphocreatine levels, because if you're doing endurance training, you really don't use that system, right? Or at least rarely, okay? So changes in ATP and phosphocreatine levels within the cell is one adaptation. We're going to look at several other adaptations in other systems. So it might not be the only one that can increase performance. Okay? Either adaptation, it would be expected to improve performance. As I said, there's not always agreement across the studies about whether or not, like, we see an improvement in training, but does that really translate to performance? There's a lot of other variables involved in performance, of course. So we would expect either of these adaptations to improve performance. That's the logical view. As I said, not all the studies support that. So we've obviously got quite a lot to learn about the first anaerobic system. Right. So system number 2 is glycolysis. Glycolysis is the system that we use to break down carbohydrates, stored carbohydrates, so glucose or glycogen. And it produces energy for anaerobic metabolism, but also for aerobic metabolism. But it doesn't use oxygen itself. It's just that the end product of glycolysis can move to aerobic metabolism. All right? So we'll see when we get to the bottom of the chain that the substrate that we end up with has a choice of 1 or 2 things that it can do depending upon how much oxygen is available to the cell. Okay? So this diagram in the textbook is quite detailed. It's got lots of very long words in it, which we don't need mostly to learn, thank goodness. If you are taking class with Dr. Barlow, you're going to have to learn it, but you don't need to learn it for me. Okay? So I'm going to make it a little bit simpler at the level that we would use it in exercise science or PE or health industries. Right? So glycolysis has 10 different reactions that are responsible for breaking down the carbohydrate into smaller and smaller chunks. Glycolysis also occurs in the sarcoplasm, in the jelly substance of the muscle cell. And each of those 10 reactions has its own enzyme that is responsible for facilitating that reaction. All right, so remember last week we said that enzymes are very picky, they only do one job, right? And they're very finicky about the temperature and the acidity level in the cell. Okay. So when we look at glycolysis, we get a little bit of ATP from a molecule of carbohydrate. Okay, so let's have a look at this. I want to do that. Okay. So the basic outline of glycolysis is like this. About halfway down the reactions, it splits in 2. Okay. So what we start off with is a 6 carbon molecule. Okay. And that is going to be either glucose or glycogen. So if it's glucose that's in the bloodstream, that has to be transported into the cell and broken down to step 1. Right? So it actually uses 1 ATP. If we're going to start off with glycogen, then glycogen, when it is broken down, happens to turn into the next step without using any energy. All right? So glucose, we have to use an ATP to get it into the chain. Glycogen, we don't have to use an ATP. All right? Halfway down, we split in two, so we end up with 2 3-carbon molecules. Okay. And we keep breaking down, we keep breaking down, we keep breaking down, and in the end, we result in pyruvate. And because we broke in 2, we have 2 molecules of pyruvate, 1 for each piece of the chain. Okay? So, let's go and fill in our ATPs. So between, oh, what happened there?
Okay.
Where'd my whiteboard go?
You're just reconnecting, I think.
Hmm?
I think you were just reconnecting.
I'm reconnecting?
What's the spinning thing at the bottom? There we go.
Okay. So, between here, where the glycogen comes in, and the point at which the 6-carbon carbohydrate breaks into 2, we use up another ATP. Okay? So, somewhere around here, doesn't matter exactly where for us. We're going to say we lose another ATP. Okay? So this is a bit weird because last week I said that carbohydrate was a great energy source because we could break it down quickly and get ATP quickly. And so far all we've done is use ATP. Right? So once it's split into the 3-carbon molecule, we get 2 ATP for each 3-carbon molecule. Okay. All right. So we make 4 altogether, and we either use 1 or 2 depending on which carbohydrate we utilize. Okay. And in the process of getting these ATPs, we also release the dreaded hydrogen. Okay. All right. So remember, hydrogen's a problem because hydrogen is acidic. All right, and our enzymes don't like it when there's acidic levels building up in the cell. All right, so we have to have something to do with this hydrogen. Okay, so we'll come to that in a minute. Okay, so my pyruvate can pick up a hydrogen and turn into lactate. All right, so first of all it turns into lactic acid. Lactic acid is highly unstable and almost immediately changes into lactate. Lactate can get dumped into the bloodstream. And when I was in undergrad, we thought it was a waste product that you had to get rid of, but now we know that lactate can be recycled by the Cori cycle and turned back into glucose and then used again for energy. So it isn't really a waste product at all. Okay. The other thing that can happen to pyruvate is if the cell has plenty of oxygen available, that pyruvate can get shuttled across to aerobic metabolism, which we'll talk about later in the week. Okay. So we've still got hydrogen floating around. Not all of it gets picked up by pyruvate and turned into lactate. What are we going to do with hydrogen? All right, we have two bus systems. Okay. We have bus number 1, which is NAD, and bus number 2, which is FAD. All right, and they can pick up the hydrogen and also shuttle it off to aerobic metabolism. Okay, so that is almost all you need to know of that whole very fancy diagram in the book. All right, so if it helps you to study using the simple version, use the simple version. If it helps you using the detailed version, use the detailed version. Okay? All right, let's go back to our PowerPoint here. So, as we saw on the diagram, depending whether it's glucose or glycogen, we may need ATP to start the glycolysis process off, right? And we generate ATP in the second half of glycolysis that we can use for muscle contraction, okay? And at the same time, we're producing these hydrogens, which are problematical because they're acidic. Okay, so a lot of the time those hydrogens get accepted, picked up by the NAD bus, so that's nicotinamide adenine dinucleotide. It's way easier to call it NAD, and if you forget what NAD stands for, look it up, right? And it can transport hydrogens to the mitochondria, which is where aerobic metabolism occurs. And we'll look at that in chapter 3. If aerobic metabolism isn't ongoing, right, it's not the predominant system at the time, pyruvate can accept hydrogen and become lactate. All right? Sometimes you'll see glycolysis called the lactate energy system. Our book doesn't call it that, right? It calls it glycolysis. Okay? So if it's pyruvate turning into lactate, that's anaerobic. At no point in that chain did we use oxygen to produce ATP. All right? So as long as the pyruvate is turning into lactate, we're still working anaerobically. We can measure that by measuring blood lactate levels. All right? So if you're in a lab, we can't do it like out on the track. But if you're in a lab, they can monitor your blood lactate levels and see if you're still working anaerobically and dumping lactate into the bloodstream. Okay. So for glycolysis to continue, it depends in part on donating these hydrogens off to the aerobic system. All right. So as However, right, if we're training an event that uses glycolysis as its predominant energy system, then we're going to see adaptations to that system to make training and performance more effective. Right? So in that chain, remember, each step of the chain has its own enzyme. So it's believed that changes in the activity level and or the amount of glycolytic enzymes would impact performance because it would increase the speed at which ATP is available using that system. Right? So we've got lots of enzymes all the way down the chain. The three that are important, and really one of them is important. Glycogen phosphorylase is at the point where glycogen is breaking down to come into the chain. Glycogen phosphorylase is facilitating that breakdown right at the top of the chain there. Right? Lactate dehydrogenase is facilitating the breakdown of pyruvate with the hydrogen and turning pyruvate into lactate. Okay? The middle one is one that you want to try to remember. It's much easier to call it PFK, all right? Phosphofructokinase. And phosphofructokinase is what we call a rate-limiting enzyme. It's in the chain around the point where it breaks into two. And so if there isn't enough PFK enzyme available, then the breaking down of the 6-carbon to the 2,3-carbon chains slows down. And remember that it's the second half where we're producing the ATP. So if that breakdown is slowing down, then the production of ATP is slowing down. So the adaptations that we see in the system are going to depend on the type of exercise that we're doing, right? Are we using this to produce carbohydrate for aerobic metabolism, or are we using carbohydrate to fuel anaerobic metabolism? Right? Okay. So some studies very frustrating. Some studies do, some studies don't, but some studies see increases in enzyme amount and activity due to weight training, sprint type training, and endurance training. Right? So there's lots of variables involved, but it's thought that that increase in enzyme activity would improve ATP availability and therefore improve performance. Questions? So in System 1 we saw enzyme adaptations. In System 2 we see enzyme adaptations. In System 1, we saw possible changes in the initial molecules that were being used, so the substrate of a kind, right? And the same occurs in System 2, in glycolysis. We see adaptations to carbohydrate storage. Okay? So, increases in glycogen storage in the muscle tissue can affect both glycolysis and aerobic production of ATP. So because it impacts aerobic production of ATP, when we do endurance training, we see increases in stored glycogen in the muscle tissue. Right? We also see increases in stored glycogen at the muscular level with certain weight and sprint activities according to some studies. Other studies say no, if you do anaerobic work, you don't see an increase in stored glycogen. It has to be endurance work to improve stored glycogen. So again, we don't have a clear answer yet on what does and doesn't improve stored glycogen. Some of the discrepancy could be the way that the studies are run and the data is collected, right? So if you're doing less than 10 seconds worth of activity for your sprint or your weight, then those studies seem to be the ones that more typically show no change in glycogen levels. Well, that would make sense, because if it's less than 10 seconds, it's not using glycolysis to make ATP. Right? If it's greater than 10 seconds, then we're starting to see studies that go, oh, okay, we see a little bit of change in glycogen levels in the muscle. All right? Because the further past 10 seconds you get, the more important glycolysis becomes until we get to 2.5, 3 minutes. Right? So it could be the length of the training program. You know, are they running the study for 6 weeks? Or are they running the study for 12 weeks or 20 weeks? Right? What kind of training are they asking the participants to do within this training program? Right? So all those kinds of things could be playing a role in why we see the discrepancy in the results. Okay. Now, unlike system number 1, we have a third type of adaptation that occurs if we train the glycolytic chain. So adaptation number 3 is changes to buffering capacity. So a buffer is the opposite of an acid. Okay? So the buffer is going to be what I would call an alkaline. I think here it's called a base molecule. All right? So it's the opposite on the pH scale to an acid. And so the idea here is, you know, if I'm producing hydrogen ions and we know those aren't beneficial, then the body is going to adapt to try to balance those hydrogen ions by having more alkaline buffer available. Right? So the longer I can buffer the hydrogen ions, the longer I can maintain force production in this system. So in skeletal muscle, there are buffers floating around in the cell, right? So certain proteins can act as buffers. Phosphate groups can act as buffers. Bicarbonate can act as a buffer. All right? So the example the book gives you is that bicarbonate combines with hydrogen and forms a weaker acid, carbonic acid, and so pH isn't affected as much as it would have been if the hydrogen was left there on its own. Right? Endurance, so aerobic and sprint training, anaerobic training, both have been seen to increase the levels of buffer type molecules available in the cell. And so that training adaptation is one of the ones that's believed to prevent fatigue in a short-term type event, right? So you train and you train and you train, and one of the things that changes is you produce more buffer substance in the muscle cell, and so the cell can keep, remember that physiological fatigue means a drop in force production. All right, so if I can delay or prevent fatigue, then I can maintain force production for longer. Okay, that means I can either go further or I can go faster. Okay. So this idea has led to the practice of bicarbonate loading that you might have heard of. Bicarbonate loading is has some problems because it can really upset your stomach and give you all kinds of gas and cramping, and some people use, not bicarbonate, they use maybe it's bicitrate, I can't remember, bicitrate. And that seems to have less effect on the digestive system. You have to take a lot of bicarbonate for it to have any effect on the levels of bicarbonate in the cell, which means you've got to really be quite careful at plotting it out and not taking too much at any one time, and taking smaller doses more regularly. I'm personally not sure that I think it's worth the risk of cramps and gas for a possible improvement. However, I'm not very competitive, so I might not be the right person to to make that choice. All right, so in system 1 we have 2 types of adaptations. In system 2 we have 3 types of adaptations. Oops, I'm missing a page. Oh no! That's not good, is it? Okay, so one of the things we tend to do at the entry level to exercise physiology is talk about the energy systems as though they are standalone systems. So I use system 1, and then I switch to system 2, and then if I keep going, I switch to system 3. That's not entirely accurate. All right, really what happens is that most of the time, all 3 systems are doing some work in producing ATP. It just depends which one is predominant for the particular event that you take part in. Okay. So, anaerobic events are high intensity, short duration, right? They rely on intramuscular stores of ATP and phosphocreatine that are already there, and carbohydrate by breaking down glucose or glycogen for energy. So things like weightlifting, shot put, high jump, diving, sprinting, right? All of those would use either ATP-PC predominantly or a combination with some more emphasis on glycolysis. All right, so if I'm a sprinter, it depends which length of race I run as to how much I use glycolysis. Right? If I'm a 100-meter sprinter and I'm good, not me, right? I'm good at 100 meters, then I only use that ATP-PC system. Only, right? It's the predominant system for that event. But if I'm a 400-meter hurdler, right? One of the reasons 400-meter hurdles is such a killer event is because it takes around 2.5 minutes or so, at which point glycolysis is running out of steam, but we haven't quite switched to aerobic metabolism. Right? That's why it's so hard. Tricky event to train for, 400 meters. Endurance events then are going to be longer duration, necessarily lower intensity, so low, moderate levels of intensity, not that really, really high, hard work. Right? So things like running a marathon, road cycles, 10K, open water swims, triathlons, all those kinds of events are endurance events. All right? But not just those events, because aerobic metabolism becomes predominant at around 3 minutes. Right? So anytime you're doing something for longer than 3 minutes, you're really predominantly using the aerobic system. Right? It just depends how hard it has to work to churn out ATP on how long am I going for. Right? So here they've produced an estimate of the percentage of anaerobic metabolism versus aerobic metabolism at certain times. Okay? So, as I said, they're both present at any one time, but as you can see here, if I'm doing an event that is 1 to 3 seconds long, then 100% of that ATP is going to be anaerobic, right? And the longer the event goes, right, the less anaerobic work is done and the more aerobic work is done until we get to around 2 hours, where if you're working at 2 hours or longer, then most all that energy is going to be coming from aerobic metabolism, unless you suddenly sprint. Right? So here's a good example. If I'm jogging along in my marathon, right, and I'm at the 2.5 hour mark, then my ATP is all aerobic. Right? And then someone comes up alongside me and starts to get past me, and I decide, ooh, hell with that, and I put on a spurt to get back past them and get back in front, then I would use some anaerobic ATP to increase the intensity to get back in front of that person. Right? But when I'm plodding along at my steady state, right, I'm keeping an eye on my time and my heart rate, that's all aerobic. Okay? So we can estimate these, and we can take these times and put them to certain events, right? And say, okay, then that means that this event is likely to be 40% anaerobic and 60% aerobic. You have to remember that there are that there are individual differences, right, in the person. There are differences between sports, okay? So you can't, it's not an absolute, it's an estimate, this chart, okay? Now, one of the useful things you can use the chart for is a principle we call specificity of exercise, right? So specificity of training. Because what you want to do is to line up your training program so that you are working the energy systems in a similar way to the way they're going to be used in the actual competition. Okay? So if I'm 100-meter sprinter, right? 90% of my ATP is going to come from my anaerobic systems, ATP-PC in particular, and only 10% of my energy is going to come from the aerobic system, then spending my time going for 3 or 5 or 10-mile runs is a bit of a waste of time, right? Because I don't use that system enough to train 5 miles, right? So I need to split my training program 90-10, right? 90% of my training session is going to be anaerobic, 10% is going to be aerobic. Does that make sense? So this is a representation of energy sources for a 3-second sprint. I'm not sure where you would use a 3-second sprint. I guess in a football game. How long does it take between bases?
About 3 seconds.
About 3 seconds. So maybe to get from the second base to the third base, or maybe a short sprint to move the ball up the field in a football game, right? So 3 seconds, we've got intramuscular ATP, so our first energy molecule provides about 32% of the energy for that sprint. The intramuscular phosphocreatine provides about 55%. Glycolysis provides about 10%, and possibly 3% aerobic metabolism, just because aerobic metabolism never turns off. It's always chugging away underneath. Right? So that gives you a good idea of the split between the systems for 3 seconds. All out, sprinting as hard as you can. Okay, that is chapter 2. So on the syllabus, I have chapter 2 again on Wednesday. So what I plan for Wednesday is to review the timeline and the major pieces of the pie for ATP, PC, and glycolysis, so that we are ready for aerobic metabolism on Friday. Okay? So what I need you to do is to review chapter 2, think about what you do and don't understand, so that I can use Wednesday to try to clear up anything that's complicated or confusing for you. You need to come prepared to ask questions if you need things cleared up, because then Friday we will start chapter 3. Okay?